Thank you for the introduction. It's a pleasure to be here today. Our committee is involved in collaborative research projects to enhance the scientific basis for the development and application of genomic methodologies for use in risk assessment. I've been asked to focus on two of our project areas today, one is on microRNA. We have two projects in that area. And the second is a new area for us, epigenetics. First, with microRNAs, they're of interest in risk assessment as potential tissue injury biomarkers. They are a type of regulatory non-coding RNA that are synthesized in a typical hairpin precursor structure that's exported from the nucleus, where it's further processed to its mature form, which is about a 22 or 23 base RNA that associates with an RNA-induced silencing complex to bind to complementary sequence and messenger transcripts and inhibit their translation or induce their degradation. So they're in general negative inhibitors of gene expression. Their interest as biomarkers is there's a couple or a small number that exhibit tissue selective expression that may fill gaps in current biomarker needs. They're pretty well conserved across species. It's very easy to make new assays. And interestingly, they're stable in biofluids because they're found in a form where they're enclosed in small vesicles like exosomes or microvesicles, or they're associated with proteins like the argonauts that are involved in the silencing complexes. So one kind of barrier to developing specific tissue injury markers where I forgot to mention there's very few right now that are known tissue selective microRNAs that are being pursued as biomarkers like miR-122 in liver. But in order to really get a full number and also look at tissues that we don't have biomarkers for, there's really a gap in knowledge in what's known for the rat micronome, which is the full content of microRNAs in an organism. The rat, which is often the toxicity model, is poorly annotated in terms of microRNAs compared to mouse or humans. In miR-base, which is the authoritative database of microRNA sequences for organisms, the current release, which is 21, there are only less than 800 confirmed microRNAs in the rat compared to close to 2,000 for mouse and more than that for humans. Also, the tissue distribution of microRNAs is not really very fully characterized. So to fill that gap, Dr. Aaron Smith at Eli Lilly proposed that they would create a rat microRNA database, or atlas, using next-generation sequencing. And then, as a committee, they would elicit help from bioinformaticists within member organizations to help analyze the data and to help come up with lists of what would be tissue-enriched microRNAs. So this atlas would help us understand better the normal distribution of microRNAs in rat and better interpretation of signatures that are seen in biofluids with tissue injury. So this is the composition of the atlas. It was done with Sprague-Dawley rats, male and female. About 23 tissues or tissue regions were selected, and small RNA was enriched from that and then deep sequenced. After analysis, the three independent groups came up with their own methods of determining tissue-enriched microRNAs. The overlap between the lists is about 165 tissue-enriched microRNAs, and depending on the method, there are more or less overlap between the lists. So in addition to making lists of tissue-enriched microRNAs, Eli Lilly identified additional over 1,000 microRNAs that are in the rat. And now we can add those numbers to what's known in mirror-based and bring them back closer to the mouse and human micronome numbers. Another analysis that was done was isomer analysis. Isomers are from the same parent gene, but they differ somewhat in length. They looked whether there was a different distribution for isomers, and the results were that they really had the same distribution as the parent mature microRNA. An issue, a well-known issue with next-gen sequencing of small RNAs is that when you make the libraries for sequencing, you have to add adapters and that act as barcodes in some areas. And these ligate on, they have some sequence bias in the efficiency of ligation. And thus, what you're measuring is quantitatively can be a little biased. So a subset of the tissue-selected microRNAs were selected for confirmation using an orthogonal method, quantitative PCR. And on the left is shown a scatter plot of the counts determined by next-gen sequencing for miR-122, the known liver specific microRNA, and much higher in liver than the other tissues are in the other corner. And then on the x-axis we have the CT values determined by PCR. And a CT and PCR, a high number is a low amount, and if it comes out early, it has a low CT. So they found that looking at these two patterns of expression, about 70% had very good correlation. So the next steps are we anticipate that later this year the atlas will be published and the data available in GEO, and which is a gene expression omnibus, and mirror-based, and that then these lists can be used to both help validate the atlas and identify potential markers for unmet biomarker needs or tissue injury biomarkers at various institutions, either using studies from samples they have in their freezers or new studies they're going to conduct. So this kind of segues into the next topic, which is measuring injury-induced microRNAs in biofluids, which can be very challenging because these can be at very, very low levels, and especially in unaffected animals. And they really challenge our current measurement technologies. Reverse transcription, quantitative PCR, is the most sensitive of the commonly used methods for measuring microRNA. But to get the increased sensitivity with that method, we often add a step called preamplification, which is a series of PCR cycles. That's just endpoint PCR, it's not quantitative. And there's concern that can add bias or variability to the data. Another question was whether it didn't matter whether you started with serum or plasma when looking at injury-induced microRNA, because them by themselves, they have very different spectra of microRNA. Another concern was how commutable are microRNA measurements. If we're really going to use them to come up with, to help validate biomarkers, we have to combine data across a lot of different sites and studies. And if you look in the literature, often results are reported CTs or relative measurements, and those are really not commutable measurements. So there's no, for example, there's no endogenous like housekeeping microRNA biofluids that people can use to normalize to. So we just formed a working group about 4 years ago that I co-chair along with Philippe Cotet. And our goal was to conduct a multi-site study using biological samples to assess the reproducibility of tissue injury-induced microRNA measurements. We would measure absolute levels, copy numbers, not relative. And the way to do that is to run in parallel calibration curves using synthetic microRNAs. So our question was, does this approach fit for purpose? Is it robust enough to use for upcoming microRNA biomarker validation efforts? So our study was designed using a model of acute cardiac injury in male Wistar rats induced by isobutyrenol, which is a very quick-acting drug. And then the study will be conducted at a central site, and then samples will be pulled from the same animals in the same treatment groups, and then aliquoted so that the same samples could be sent out to multiple sites. And the sites would be asked to use a standard protocol to analyze the samples. And the standard protocol was designed by consensus for the use of instruments that all sites would have available. For instance, 4 years ago, digital PCR was not really in common use at all sites. So we decided to measure 4 microRNAs by absolute quantitation per sample by RT-PCR, and the 4 are based on literature reports. 3 of them are, there was evidence that they increase in biofluids serum with cardiac injury. It's MIR1, which is the most abundant microRNA in the heart. But it's also in other muscle. MIR208, which is one of the few known cardiac selective microRNAs, and MIR499, which is reported to be enriched in cardiac tissue. We also chose a reference microRNA, one that we expected to be easily measured in biofluids, but not necessarily affected by treatment, and which is MIR16 and MIR192, depending on what one of our studies. And we also looked at a spike in control and a Arabidopsis microRNA. So our study had 4 phases. One was the dose selection of isobutyrenol. It was based on the gold standard of troponin induction at 4 and 24 hours, and a moderate amount of cardiac pathology at 48 hours, and of course survivability. After selecting the dose, we first conducted a study that compared serum and plasma from each blood sample. We split it into 2 and either processed it for serum or plasma, then combined these across animals in the 2 treatment groups. And the blood was collected 4 hours after a single dose of isobutyrenol. The third phase involved comparing plasma collected at 24 hours after the single dose of isobutyrenol, or urine collected from 6 to 24 hours from the same animals. And finally, the last phase, we kind of focused on a couple steps in the protocol that we thought were a potential source of variability, and also looked at best practices for determining the lower limit, where the data, the border where it's reliable or not reliable. And for this phase, we didn't use biological samples. We used calibration curves from synthetic microRNAs using different multiplex protocols. And so one of the first issues we discovered when looking at our data was that when we diluted down our calibration curves to the levels we needed to see baseline levels of some of our microRNAs, the calibration curves became nonlinear, and we also had a value in our no template control, which should be our blank. And this is the kind of phenomena that can be induced by or replicated if you have a small amount of contaminant. But in this case, we actually investigated it quite thoroughly, and it was due to generation of a PCR artifact, a primer dimer, that is a result of certain specific combination, protocol and reagent combinations. So this was kind of inherent in our data, so we had to deal with it. So how do we estimate values for unknowns or determine our limit of reliability with this kind of nonlinear calibration curves. And normally for PCR, you expect like a doubling per cycle. You expect it to be very linear until you get no detection. So our first instinct was just to use the linear portion of the curve and throw away the rest of the data. But when we consulted with a biostatistician at SAS Institute, he doesn't like to throw away data, so he advised us to fit all the data to a model, a 3-parameter logistic model that kind of the examples on the left there of fitting all the data. And then by inverse regression, using our unknowns for all, which we just have a CT, we want to determine the copy number by inverse regression. His model also gives you a confidence interval around each unknown calculation. So you can see that our plasma levels are in the more linear part of the curve. We had pretty tight confidence intervals. But when we looked, and for this example, the urine levels were much lower, so they have pretty wide confidence intervals based on the calibration curve. So our next step after we were able to determine values for all of our unknowns was determine like what's the demarcation between reliable and unreliable data. So we've been working on developing decision trees for the lower limit of quantitation that actually works with our data. We have over 60 calibration curves calculated with our phase 4, so we can run all these through our models and make sure we can, our models account for all the types of data possible. So I don't expect you to read what's in the boxes, but one of the models is based on thresholds that are determined from the 3-parameter logistic fit, the error that's around each measurement, especially at the lower end of the curve, the lower concentration part of the curve. We can set a threshold for how much error we allow around each measurement, and that's one method. The other defines the no template control, the blank value, as our baseline noise, and then takes a step back from that to give you kind of a conservative estimate of reliable data. So this is some of the data plotted from our study, and our whole point is really to be able to combine data across sites and independent studies and have be able to get biology out of it, not just technical noise. So starting from the left, MIR1, that's the microRNA that's abundant in heart, but it's also in muscle. So you find pretty good baseline levels in serum or plasma. And the copy number per microliters is what's being plotted. So we could see similar levels in serum and plasma, and at 4-hour plasma and 24-hour plasma, and a nice treatment effect that was pretty much the same between serum and plasma, and started to decrease a bit at 24 hours compared to 4 hours. We applied to this data one of our methods for determining the limit of detection, and when we look at our next microRNA 208, that most of the values in the control animals were below our reliable range, but we got a nice induction at 4 and 24 hours. So if you can live with having unreliable data or no, kind of blank data in the controls, that seems like a good biomarker. MIR 499 was less reliably detected in control and treated, so it looked less promising, and a treatment effect was not apparent in all samples. And the MIR 16, good agreement between sites and similar between plasma and serum, and same with 192. So in summary, although we can see similar levels of injury-induced microRNAs across sites, it's not easy, and I think some of our current methods are really not fully optimal to get the sensitivity we need for going further with testing candidate microRNAs. We didn't see any difference between serum and plasma with the cardiac injury-induced microRNAs, which was encouraging. Collecting urine over 18-hour period was not really effective for seeing any potentially seeing any cardiac-induced microRNAs in urine. Perhaps doing shorter collection time could be a choice. And the preamplification step did seem to be problematic in adding variability depending on certain protocol and reagent choices. So the lessons we learned in this project obviously are going to be very valuable when we do try to go forward with potentially validating some of the results with the rat atlas. So now the last part of my talk is on our new project area in epigenetics. There's been a lot of new data in this field because of the advent of next-gen sequencing. So we thought it was time to first review the state of the science for its use in risk assessment, and whether there was any specific questions that we could pursue in the form of a new project within our committee. So we held a symposium about a year and a half ago to look at some of these factors. So this is an incredibly brief review of epigenetics, but just some main points is that we're referring to heritable changes in whether a gene is turned off or on. That does not involve the underlying sequence. And the marks that determine this are two flavors. One is DNA methylation, and the second is really a variety of histone modifications. So another area that's kind of opening up is that there's more appreciation that a number of human disorders besides cancer may involve epigenetic changes, including, like, for instance, metabolic disease. So there's more interest in developing drugs in this area beyond oncology. So one of the questions is, when you have potentially more exposure to epigenetic modifying drugs and also to environmental chemicals which can act the same, act through epigenetic mechanisms, these can have potential adverse effects on future generations that are not exposed. So these are called transgenerational effects. So how do we measure this, or at least try to get a handle on this? Rodent assays are very expensive. They take a lot of time and resources. So a potential, proposed a new model is zebrafish. There's the reasons for looking at it. There's strong conservation of gene and protein structure and function with humans. They're easy to manipulate genetically, and they have a relatively short time of breeding and development. And there's some examples in the literature of looking at environmental agents and with zebrafish as a model for transgenerational effects. So this project was brought to our committee by Mike Lawton and Bob Chapin at Pfizer. The goal is to conduct a transgenerational study in zebrafish using a epigenetic modifying tool compound. In particular, this targets a specific enzyme that causes a specific type of histone modification. The goals are twofold, both to look at zebrafish as a model system and also to look at the sensitivity and reliability of methods for measuring different epigenetic parameters, which are more used, I guess, in academic research than in regulatory settings. So this is a model of the study that's being conducted. The F0 generation, which is the mature zebrafish, are being treated with the tool compound for 4 weeks. It's being administered orally in a clam-flavored gelatin. And then after 4-week recovery, a subset of the fish will be, or are being sacrificed for the endpoints of histopathology, gene expression using RNA-seq, and histone methylation, which is the drug target. The rest will be mated to produce the F1 generation. which will be allowed to mature and then mated to produce the F2 generation, which is the first generation you can establish transgenerational effects because the F1 generation can be exposed in its parent as a germ cell. And then those will be allowed to and then analyzed for the same endpoints I just measured, as well as drug levels in the FO generation, histomethylation in whole fish by IHC, global DNA methylation, and perhaps ChIP-seq. Certain tissues are listed on the slide that will be looked at specifically in the whole fish, and the different endpoints are going to be analyzed at the US EPA, the University in Amsterdam, UPenn, as well as at Pfizer. And additional assay, the FO generation, is going to be subject to what's called a fin clip procedure. That's a way of looking at the effect of drug on the somatic stem cells, because you get generation to replace the missing fin. It's just the stem cells are stimulated. And then you can just look at the morphology of what grows back. And this study is going to be run in parallel at the U.S. Army using additional reference compounds and embryo exposures. So at the end, we hope to have a better assessment of zebrafish as a model, some idea of whether this tool compound can induce transgenerational effects, and which of these epigenetic endpoints were more useful for assessing these potentially adverse transgenerational effects. And this project is relevant to both the pharmaceutical and environmental exposure areas. So I want to end by showing on the slide our leadership team, my co-chair, Heidrun Ellinger at Bayer, our program manager, Reagan, and her associate, Melissa, are part of the team. And finally, the member companies in our genomics committee. I'm listing here the affiliations of our members. So I'll be glad to take any questions at this point.
The last model with the hypermethylation, a really interesting model of the epigenetic mechanism. Could you also target changes due to the histone complex in the zebrafish?
Look at changes to the histone complex. Yeah. I think they're not getting in really deep mechanistically in this study. It's but they've already established that the drug does work on the fish in terms of affecting a certain type of methylation of histones. They haven't looked at the complex. I think that'd be part if they do the ChIP-seq, where you pull down certain complexes and look at the sequence that it's bound to, comparing treated, non-treated, that might get at some of the complexes that are altered by treatment.
Yeah, hi.
Can I just ask you, if you take the last zebrafish study, and you complete the study successfully, what species will you be intending to translate the information to?
Well, that's going to be kind of, I guess, that's the question, I guess. I think we're thinking at first it's just a phenomenon, and then it would really have to show the relevance to humans. That's not going to be easy. But I think now it's just looking at the phenomenon.
The principle, I'm trying to get at the philosophy though. I mean, it's very difficult to extrapolate a mouse data to humans or a rat data to humans if you look at end effects. So I'm just wondering if you do a zebrafish, whether you're likely to be moving in the direction of being more likely to translate your data to humans than you would be from a rodent or from a dog, or in other words, is there a philosophy that I'm missing as to why this?
No, I think it's just an easy way to start out. There probably would be some in between studies, animals chosen. I don't know the whole paradigm of further testing, but I think you would then proceed with some other model system to validate the results, and maybe an animal that's more used in regulatory testing.
Yeah, and also not to mention that you can do multi-generational
Yeah.
studies in a short period of time.
Yeah, it's a good screen.
To me,
It's-
that's
yeah.
just the selling point of going something like zero patient.
Yeah.
In your discussion of the microRNAs, you indicated that the database was going to be built on the Sprague-Dawley rat, but then I thought in the collaborative study you indicated that phase 3 was going to be done with the Wistar rat. Are there
these are kind of
strain
independent.
relation?
These are kind of independent studies. The Atlas was built on Sprague-Dawley, and we don't know the differences, I guess, between strains.
Okay.
There may not be that much difference, but the methods validation or the methods assessment was done in Wistar's because they were more familiar with what isopaterenol would do in that model in terms of choosing the doses based on all the troponin work.
Okay, in the epigenetics part of your presentation, I may have missed it. You referred to the tool compounds. What are the tool compounds?
It's proprietary, the tool compound. It's a Pfizer compound that at some point we'll know about, but not now.